Hi, my name is Marom Bikson, and I want to talk a little bit about how low of an electric field can actually affect the nervous system. And this specifically as it affects things like neuromodulation and transcranial electrical stimulation. Now, I think this is an equation that is very useful to answer this question. And in general, explain the effects of brain stimulation, electrical stimulation, neuromodulation technology. So it has three terms. The first term is VTM. This is the amount of transmembrane polarization, VTM, produced in a neuron during electrical stimulation, or specifically in a compartment of a neuron, like the soma of a neuron, the dendrite of a neuron. And when you're thinking about neuromodulation, that is the first mechanism of action. Everything else is derivative to that. So that's the first term. On the other end of the equation is EF, which is the electric field. This is the intensity of voltage change that exists around this neuron or around this neural element. And all forms of neuromodulation produce a certain intensity electric field in different parts of the brain. So that is what the neurons are exposed to. And then in the middle is this other term, this A or coupling constant that says for a given intensity electric field, how much transmembrane polarization do you get? And it's just a number, it's a constant. So for example, if you produce an electric field that is 1 millivolt per millimeter, and the coupling constant is 1, then the resulting polarization is going to be, the coupling constant is 1 millivolt per millimeter, then the resulting transmembrane polarization is going to be 1 millivolt. And the thing to keep in mind is that this is per compartment. So in electrical stimulation, the whole neuron doesn't polarize the same. You may have one end that's hyperpolarized, one end that's depolarized, another part that's not polarized at all. So this equation applies to a compartment of the neuron. And this equation really tells us a lot. It tells us that if you have more electric field, you're going to produce more transmembrane polarization. If you have 100 times the electric field, you're going to produce 100 times the transmembrane polarization. Now, if you produce enough transmembrane polarization, you start activating secondary nonlinear membrane conductances and then action potentials. So other things can follow that will produce different changes in transmembrane potential. But this is the expression that tells you how that first coupling happens. And different neurons and different compartments within each neuron will have different coupling constants. So a lot of times we say, for example, that an axon terminal may have a rather large coupling constant compared to a soma, and that's why it might be polarized more. Now, this equation has been worked out extensively and over years, and I'm giving a bunch of references here based on PMID. It's been shown to be more or less correct for spheroids. It's been more or less correct for when you have an axon in a transverse passing electric field, which is not the typical form of stimulation for axons. It's been shown by my lab and others to be the case for axons that are sort of infinite and then they suddenly terminate, or axons that make a very sudden bend. They will polarize across this, according to this rule. It's also been the case to show that this is correct for compact neurons. So this is actually many neurons in our brains are compact enough for this to be the case. We've shown that for cortex and hippocampus. It's also been derived, for example, using something called the mirror estimate. And again, I'm going to refer you to these references for people who really want to dig in. It's not always the case. So it's sometimes the case that you have non-compact structures, axons of passage. And in those cases, we cannot think of the electric field, but we must think of the change in the electric field. And why this is pretty canonical in a lot of sort of neuromodulation modeling, and practically speaking, we can often just get away with considering the electric field. And this is then something I call the quasi-uniform assumption. The quasi-uniform assumption just says you go to a part of the brain, you look at the electric field and assume that it's one value around a neuron, and that is then the value that you apply for this equation. And it's reasonable under many situations. But again, to be very clear, there is more complicated math when you have non-compact structures or structures unlike what I'm talking about here. And so when you look at this equation and you say, well, then what is the lowest possible electric field that will have an effect? The answer is there is no lowest electric field, right? Because this is linear. No matter how low the EF is on that one side, you're going to get some VTM, right? So there's no electric field at which point you get no polarization at all. And then you may also say, well, because transmembrane polarization in neurons has such an important role in neuronal function, therefore, someone might speculate, there is no minimum electric field that is inert. No matter how weakly we stimulate, no matter how weak electric fields we're exposed to, they may always have the possibility of having an effect. And again, this is a question that we and others have looked at, where you really start to think about, well, how small of an electric field can you demonstrate has an effect? But I think this way of thinking is as I'll talk about next, can be illustrative, but also a little bit misleading, because you get into this how low can you go game. This is basically studies in animal models and tissue where people try to demonstrate what is the tiniest possible electric field that will still have an effect. These are the kind of experiments that you'll see. So for example, in an animal model, you can apply the same electric field over and over and over and over. So you're applying it hundreds, thousands of times and trying to measure very, very minute changes in neuronal function. And so again, we and others have done that. It's an experimental system. And so you can design these experiments to be very, very powerful. And you can resolve things like in this paper that fields as low as 0.2 volt per meter, which is the same thing as 0.2 millivolt per millimeter, could have, in this case, have an effect. But you can see now how this can be, again, needs to be carefully interpreted because of this notion that this is a controlled experimental system, and we can specifically design experiments to pull out these very, very tiny, statistically significant, but very, very tiny effects. And that may not really be the question that we want to answer when you're trying to understand what is the lowest electric field that would be meaningfully important in the example of, electrical stimulation. And I should also mention, because this is sort of a statistical game, it's also possible to design experiments where you don't see effects. I could deliberately design an experiment where a relatively large electric field did not show an effect. And again, I don't think that would be any more illustrative than going the other way. We're not looking for a single electric field that is the magic threshold. I think what instead we're looking for from animal studies that measure the effects of low electric fields is explanations that help us understand how electrical stimulation, and particularly low intensity electrical stimulation, like TES, transcranial electrical stimulation, TDCS, and so on, has an effect. And that's where I think these kind of considerations start to matter. And so we can ask questions like, can an electric field that is too weak to produce an action potential, right, where the VTM is too small of a change to produce an action potential, still profoundly change the function of the neuron? And we and others have explained how that is. We can ask questions like, what about oscillation? So these are already emergent network phenomenon in different parts of the brain. Are they especially sensitive to low intensity electric fields and high intensity electric fields? And why? What is the explanation for why they're sensitive? And these kind of studies can address that. Is plasticity affected by stimulation? And why? What is the explanation for why ongoing plasticity in the brain is so sensitive to electrical stimulation? What about other cell types? I've been very interested in endothelial cells. People study glial cells. Is there an explanation for how electric fields might affect them and how their dose response, their sensitivity may be others? We can ask questions like, is more always more? Right? It's increasing the intensity of stimulation, giving you a bigger electric field, even though that will produce a bigger VTM. Will that always translate into a bigger outcome as far as what we're interested in? We can also ask questions like, there are examples where we see polarization that seems bigger than what this equation would predict. This sort of superlinear sensitivity that may result from cellular properties, network properties. And again, animal models can help us understand where the super sensitivity might come from, which is interesting because it tells you that when you have low electric fields, you may be looking for these super sensitive compartments. And as I already mentioned, looking at sort of other neuronal cell types. So I've given you this equation. I think this equation is a wonderful starting point. But I guess what I'm trying to say, it's not the ending point. Okay.